So now we have the pleasure of hearing Anil, who did his project with Moshe.
Good afternoon to all. I am Anil from Nepal. I did my research exercise with Professor Moshe Kol in insect ecology lab. My topics of the research exercise was pollen consumption by field population of the 7 spotted ladybird beetles Coccinella septempunctata. This 7 spotted ladybird beetles is dominant aphidophagous beetles. It feeds on the aphid, not only the adult, the larva also feeds on the aphid, and they consume lot of aphid in their life cycle, and it is introduced and well established in different part of the world. These beetles utilize different kinds of the habitat and different kinds of the agroecological system. It is well established in annual ecosystem, perennial agroecosystem, and in the different different climatic conditions also, and it is hardy in that cases. But it is also invasive outside its native range. Although it is beneficial in the native range, in some cases it is invasive. In North America, it has been found that upon its successful establishment, it replaced the native 9 spotted ladybird beetles, which is Coccinella novem notata, and it is zoophytophagous facultative omnivore. The food of these beetles consists of 2 types. Conventionally, the food was divided like this: essential food. The essential food ensures its completion of larval development and oviposition by the adult, whereas the alternative foods are those food which provide the energy source, and they prolong the survival of these beetles. And the essential food of these beetles consists of 24 species of aphid. Regarding the essential food, it is considered as quite specific compared to the other coccinellids, whereas in the case of alternative food items, in its food it consists of fungal spores, thrips, other non-aphid arthropods, pollen, and considered as somehow urophagy or generalist predator. Like the generalist predator like Coccinella septempunctata and other predator, it is very important to know the role of the plant provided food item. So, plant provided food item the role of the plant provided food item has been explored a lot. Out of them, let us talk about some of the roles of the plant provided food item. And these plant provided food items support survival in the absence of prey, or when the prey is of inferior quality compared to their principal prey, and these plant provided food items act as a fuel for the migration to go to the other area. When there is no prey, they will incorporate those plant provided food materials like nectar and pollen, and they will get energy from that. They will reserve energy and they travel to the long distance. And they are also found to improve reproductive capacity, and also has to found to enhance their dispersal after the diapause stage and quiescent and overwintering period. These plant provided food materials also help to introduce intra-guild predation, and also they reduce cannibalism. It is one of the cannibalistic species. It would feed on the egg of its own when they don't find on other food materials. If there is the food materials like plant provided food materials, they would not feed on their egg. But there is always a trade-off between consumption of plant provided food materials at a specific period of time and the consumption of the prey, because plant provided food material itself is a alternative to the prey, and they would distract feeding on the prey, and there will be less suppression of the prey for the particular period of time if we consider only for the short period of time. So, among the plant provided food materials, pollen is frequently associated plant provided food materials for this beetle. In the field, it has been found that they are aggregated near the flowering area, and they are feeding on the pollens, and also during the gut dissection done in the different research, they have revealed that almost in 80% cases these pollens are frequently occurring in the gut of the insect. That reveals that these are important different plant provided food materials for these beetles, and it has been found that they provided the fitness benefit in the field and also increased the fecundity in the lab condition. Compared to the other coccinellid beetle, Hippodamia variegata, the research done in our lab, the benefit, if we consider also the prey suppression, the mix prey and pollen mix benefit was greater to this Coccinella septem punctata compared to the Hippodamia variegata. But we found, based on the current research and other findings, we found there is not much more exploration of this field behavior of these beetles, because it is important beetles for the field condition as well as in laboratory condition. We thought that maybe we can contribute somehow to the pollen feeding behavior in the field condition, and maybe in the male and female there may be the different kinds of pollen feeding behavior, because we thought because male and female has different reproductive role and they need the different nutrients in different amount, and we hypothesized that male and female of this beetle utilize pollen in the same way. Maybe they utilize the pollen in different way. Another hypothesis we put forward was that pollen of different plant species are consumed according to their relative abundance in the habitat. Maybe the pollen of different plant species are consumed differently by the beetles regardless of their abundance according to their preference. For this, our objectives was find out the pollen consumption behavior of ladybird beetles collected from their natural habitats, and specific objectives were quantify and compare number, diversity and richness of pollen consumed by male and female population of these ladybird beetles, and another specific objective was find out the relationship between proportion of the flower present in the habitats and their relative abundance in the gut. And for this we selected 5 different location: Mismar David, Kefar Menahem, Peth Gobrin, Park Hamda and Nest Joina. Within those locations we have different kinds of habitat. There we can see crop field with trifolium, herbaceous field, crop field with wheat, and crop field with kohlrabi and cabbage, and also within the herbaceous habitat also there is somehow difference regarding the plant composition. In some of the herbaceous field we can see in this dry herbaceous there is less amount of flowering plant, whereas in the case of flowering herbaceous there are more flowering plant, and the denotation and acronyms will be later will be used like this. Mismar David I will use MD, if it is trifolium field MDTF, and like in the herbaceous field of Kefar Menam I will use KMS. These are used in later. And materials and method, we did flower sampling and flowering area measurement from where we collected the beetles, and then we collected the flower from those area and nearby area, and we prepared the reference pollen, and we characterized those reference pollen based upon some of the morphological characters of the pollen, like pollen dispersal unit, size, shape, exine ornamentation and coloration. And we did beetle sampling in between 4 April 2017 and 19 April 2017 from those habitat. And before doing gut dissection we did sexing of the beetles, because we were interested to see the pollen feeding behavior between the male and female. And we did the identification of the pollen to the genus level, we did by the use of microscope. It is on the 400 magnification. And our results are like this. The amount of pollen consumed by male and female ladybird beetles in the field. Before comparing the amount of pollen consumed by male and female in the field, I did some correction for the body weight of the male and female. Female of the Coccinella septem punctata are larger than that of the male. So we did correction for that body weight, and we found that in whole population of 46 females and considering the 37 male, female consumed more amount of the pollen that of the male. It was almost 29% higher than that of the male, but we also did separately the comparison of the pollen fed by the female and male in the different habitat. Here we among the different habitat, only we in the Mismar trifolium field of Mismar David habitat, there was significant difference in the amount of pollen consumption by the females than that of the male. But in all cases, female consumed more amount of pollen than the male. I already said that female has different reproductive roles than that of the male. They need more amount of protein, and pollen contains good amount of the protein. That might be the reason that they incorporated more amount of pollen than that of the male. And we also did the diversity index comparison of the pollen diversity index consumed by female and male. We were interested to do this because whether male or female incorporate the pollens from different plant taxa, more different plant taxa, and for this we calculated first Shannon-Weaver diversity index of pollen for each beetle, and then we compared the diversity index of pollen between male and female. We found that the diversity index of pollen consumed by beetle was female beetle was significantly higher than that of the male beetle. Similarly, the richness of the pollen consumed by the beetles, female beetles, was significantly higher than that of the male. And we thought that maybe female needs different due to different nutritional need. They might have incorporated pollen from different plant taxa. They can. And also we were interested not only in the sex of the beetles, also we were interested to the different habitat, because habitat management is important in the case of conservation biological control. And we found that there is also significant difference regarding the species richness of pollen consumed by the different beetle from the different habitat. We found that in the herbaceous habitat of Kephar menaham, the specific richness of the pollen was higher, significantly higher than that of the trifolium field of Mismar David, herbaceous habitat of Beth Gobrin, dry herbaceous habitat of Nes Zoena, kohlrabi and cabbage field of Nes Zoena, herbaceous habitat of Park Hamda, and trifolium field of the Beth Gobrin. And we can see some of the interesting result that normally we can see that herbaceous habitat are having more pollen richness, but in some cases you can see wet field of the Kephar menaham, there is also good amount of pollen richness as compared to the some of the other habitat following this habitat. So and we were interested what is this, what is the reason behind this, and we that was that this wet field of Kephar menaham and herbaceous habitat of Kephar menaham were surrounded by the shrubland. And there was shrubland nearby with 8 different kinds of the flowering habitat in that area. Maybe they did the offsite utilization of the pollen resources from that area. It is also found in other insect also. In the small bee, it was also found that offsite utilization of the resources apart from the conventional field. And we also did the amount of pollen consumed by the beetle from the different taxa in the different habitat. We found the significant result almost in all cases, but maybe the taxa is different in the different habitat. We can see in the herbaceous habitat of Kephar menaham as well as wet habitat of Kephar menaham, they consumed more amount of pollen from the this Sinapis species, here compared to the Notobasis, and here compared to the Triticum aestivum, wheat, Centaura species, and Amnimazus species. So they consumed more amount of pollen from the Sinapis species. Similarly, in the case of herbaceous habitat of Park Hamda, they consumed more amount of pollen from Sinapis species and Chrysanthemum species as compared to the Centaura, Acacia, and grass species. In the case of kohlrabi habitat of Nes Zoena, they consumed more amount of pollen from Raphanus species, Polygonum species as compared to the Acacia species. Whereas in the case of dry herbaceous habitat of Nes Zoena, they consumed more amount of the pollen from Polygonum as compared to the Right. Polygonum as compared to the Sinapis and Raphanus. In the case of flowering herbaceous habitat of Neszoena also, they consume more amount of pollen from Polygonum species compared to the Sinapis and Pinus species. Then we were interested to see which kinds of the habitat provide more proportion of the pollen to the beetles, and is there some kind of trend regarding the providence of pollens from the habitat. We can see that from the herbaceous habitat of Park Hamda, almost 99% of the pollen is pollen out of the total amount of the pollen consumption is provided. Similarly, the good amount of proportion of the pollen is also provided from the herbaceous habitat of Beth Gobrin, but we can see from the wheat habitat of Kephar Menam and dry herbaceous habitat of Neszoena there is less amount of pollen provision. But interestingly, although it is a crop field, we can see in the Mismar trifolium field of the Mismar Devi, they are providing good amount of the pollens from the habitat. Maybe they might have preferred the pollen from the trifolium field, so there is good contribution from the trifolium field for those beetles. Then after we were interested to see the trend of the flowering percentage in the habitat and the provision of the pollen from those flowering plant, and we can see that in the herbaceous habitat of Park Hamda almost 40% flowering area is contributing 99% of the pollen consumed by the beetle, whereas it is also the herbaceous field of the Kephar Menam, but the relative amount of provision is less as compared to the herbaceous habitat of Park Hamda, and it may be due to that because the herbaceous habitat of Kephar Menam they might have utilized the pollen from nearby habitat, whereas in the herbaceous habitat of Park Hamda they consumed more from that field, and because that habitat was also surrounded by the human settlement and there was no more choice for them. And we can see in the wheat condition more flowering percentage of the wheat is contributing less amount of pollen consumed by the beetle out of the total amount of pollen, whereas in the case of trifolium field less amount of flowering proportion of the less percentage of the flowering area is contributing more percentage of the pollen consumed by the beetles. And at last we were interested to see the relationship between percentage occurrence of various flower in the field and their presence of the pollen from those flowers in the gut, and we found interesting relationship visual impression from that. We can see in the case of Sinapis arvensis, in most of the case the less percentage occurrence in the field is also contributing more percentage of their pollen in the gut. Although even in the case of herbaceous habitat of Kephar Menam, you can see we cannot see the Sinapis in the flowering in that area in the habitat from where we collected the beetles, but they incorporated the Sinapis pollen from the nearby habitat. So in the case of Xylobium marianum, we can see that more proportion of the flowering was in the habitat, but they incorporated less proportion of the pollen out of the total pollen consumed by the beetle. Similar phenomena we can see in the Chrysanthemum coronarium. The more percentage of the flowering in the habitat is contributing less proportion of the beetles in the gut of the beetles, less proportion of the pollen in the gut of the beetles. So this result this pattern of the occurrence of differential occurrence of the flower in the habitat and the pollens in the gut suggests that there is there is no straight relationship between the occurrence of the flower in the field and their occurrence percentage occurrence in the gut. So it can suggest us their preferential to some pollens, but to explore in it more we have to do the research in the landscape level, because I did it in the habitat level, and so that will help to make more general recommendation. So from this research I concluded that pollen consumption by male and female is comparable, but in some habitat female consumed more amount of pollen. Diversity index and species richness of pollen consumed by female is always higher than that of the male. Amount of pollen consumed by ladybird population from different taxa is significantly different in different habitats, and species richness of pollen is higher in the beetles collected from those habitats where nearby habitats are herbaceous or shrubland, and relation between proportion of the pollen from different taxa in the field and those present in the gut is different for different taxa. It is not the ratio of the available of flowering proportion in the habitat and their availability in the gut is not constant. There is differential relationship between their presence in the habitat and their in the gut. So I want to acknowledge my scholarship provider GeneCo2 Foundation Canada, similarly the PS Foundation, and then I am grateful to the International School of Agriculture Science, and here the team academic head Professor Moshe Cole, plant science head Alon Samaj, environmental health head, as well as other management team Nurit, Lirit, and I am thankful to my mentor Professor Moshe Cole and my supervisor Sakha in our lab and our technician Ruthi for their support always, and I want to say thank you to all for listening this presentation. Questions?
What was the physical size of a sampling site for each of your sites?
For the flowering area measurement?
For the pollen, with regards to determining if it's in the site or from the surrounding habitat site.
Size of the habitat?
Yeah.
It was different for the different cases, because if it was a crop field, it depends on their availability in the field.
Okay, and so based on any of the given sizes, is it normal for a beetle to fly a greater distance in its general foraging patterns than the size of the sampling site?
Can you please repeat
Meaning,
it?
is it normal for the beetle to fly a greater distance than the size of the site that you would look at?
No, normally we focus now in the habitat level, but we also have the information about the area where within the 300 meter, but from our research, and it has been shown that they have also incorporated pollen from the larger distance also. From the previous research, it has been found that they have also incorporated pollen from the 73 meter in the case of the canola pollen. But in our research also we frequently found that they incorporated pollen from pine, which was not there. Maybe pine was blown by the wind, or they also incorporate from larger distance or larger distance. So they can incorporate.
Do you think that there are any preferences for the beetle in how it chooses the pollen? That many beetles may like one kind of pollen or another kind? Do they have any attraction to any kind of pollen? Because when you look at the results, you don't see something like this. It's not that in all fields they like the same flower.
No, yes, it was different.
But if you check it in vitro, do you think that you will have some kind of preference towards one kind of pollen?
I think they may have some kinds of preference, in, so we were also hypothesized for that. And but also I also read some of the papers regarding other insect also. They have written that in some of the cases, before dismantling the pollen, they cannot assess the nutrition from the pollen. So there may not be that they cannot assess the nutrition, one of the hypothesis. And other also from our pattern also, they also incorporate the pollen from the larger distance where they were not available in the cases. So they have the preference, I think.
Why do you think that the females prefer more pollen than the males?
I think it is mainly based on their nutritional need, because female need pollen mainly protein, mainly in their ovary development and ovarian follicle deposition. So they might have incorporated for the more amount of protein.
How many ducts did you open?
We opened 89 guts. Out of them, 6 guts were empty, there were no pollen, and we studied for the 83 guts with the pollen. Out of them, 46 were female and 37 were male.
And when you talk about the area of each plant, right, you have the pollen area and also
The flowering area covered by the each
flower
plant.
area.
Yes.
Do you have to take into account how much pollen is made per area, or because each plant makes a different amount of pollen?
Yes, each plant may make different amount of pollen, but for this time we mainly consider the flowering area, especially the flower area, which is responsible for the pollen bearing flower area. For example, some of the flowers have small area and some of the flowers have large area. We assume that the pollens from the large flower is more than compared to the pollen from the small flower. So we basically measure the area of the flower, considering that the pollens from that large flower is more than that of the small flower. There.
You said that females require more protein for its reproductive activity, and I think you measured this beetles in the season. Did you try to see the same results in off season, like when there is no reproductive activity?
I already mentioned that we did this sampling from April 4 to 19. It represents different. It does not represent different season. It is the specific period of the time, and so I cannot say based on that.
Okay, thank you very much. Okay, so our next up will be Marita, who's did her work under my supervision. So please.
Yom Tov, I'm Milica Perisic, and for the past year I've been working in Professor Alan Samach lab on the topic of temperature induction of flowering in olives, and more specifically at what time during the day should we measure flowering induction parameters in olive. So olive, or Olea europea, is a small evergreen tree species that grows in countries of Mediterranean region and on some other continents and countries like Argentina and California and Saudi Arabia. Now, in all these countries, olive has a major economic significance, but it is also more than that. There is a very famous Greek myth that speaks about how Greek goddess of wisdom, Athens, won a patronship over Greek polis of Athens by giving Greek people a gift of olive as a plant. And Athens is, after all, the symbol of the civilization. Olive has been important enough to be first plant to have written instructions about its growing specifics about olive growing, some 350 years BC, if I'm not wrong. And main points haven't changed much since. So olive flowers in April, and the fruit sets and develops and ripens on the trees from May until the harvest, which happens from September to November, depending on the cultivar. Now, what we can see as flowers in April is the consequence of flowering meristems being formed in the second half of the February. On the tree, on this picture here, you can see. you can see the picture of meristem with clearly visible signs of morphological signs of inflorescence being formed in February. So what causes the transition of the meristem from vegetative to flowering meristem? Now we know that in Arabidopsis, under inductive conditions, which is in Arabidopsis long days, there is a specific gene called flowering locus T, whose transcription is upregulated, and FT protein travels from leaves through the phloem to the shoot apical meristem, where it upregulates some other genes that are involved in transitions from vegetative to flowering meristem. Now, for the past few years, a lot of work in Professor Alan Samach's lab has been focused on olive, and they have identified two FT-like genes in olive, naming them OE-FT1 and OE-FT2 after Oleo europea. They have monitored these genes in various conditions and throughout the year, and they have seen that their expression is baseline and very low throughout the year, throughout the summer, and it starts increasing in December, and it reaches peak in January, in late January, which correlates with flowering, with inflorescence initiation in the February. Now, that led them to the hypothesis that low temperatures during the winter might be responsible for flowering initiation in February. So how can they be sure that these low temperatures are what causes flowering initiation and not some other factor? Well, they have also monitored flowering rate and have seen that trees that were exposed to the period of low temperatures during the winter flower profusely, while trees that do not have this cold period do not flower at all. And at the same time, the expression of these FT genes in olive is upregulated in the trees that have this period of low temperatures, while in the trees that do not flower and did not underwent these low temperatures is not upregulated. Now, everything up to now seems to work in the favor of their hypothesis, but could there be a problem with this story? Well, possibly. We know that in Arabidopsis, FT transcription is upregulated by constans protein, and constans protein expression is controlled by circadian clock, and the expression of constans protein has two peaks in early morning and late afternoon. But the constans protein itself accumulates to a sufficient level only in the late afternoon because it's stabilized by the light. So circadian clock that controls the expression of constans protein is internal mechanism that helps plants and all other organisms to keep in track with the time. In other words, to anticipate the daily changes in light and temperatures by changing the level of expressions of certain genes that control certain physiological processes during the day. The output of circadian clock is composed of the gene whose RNA expression oscillates during the day as a response to the oscillations of expression of the genes that compose the central oscillator. Now, when we experience jet lag, that's our circadian clock being entrained by locally environmental clues, which is in mammals, in humans, and also in plants, light and temperature. These two genes, LHY and Gigantea or GI, are part of circadian clock, genes that are part of circadian clock, and you can see here that they have clear diurnal expression. Light and dark bands on the horizontal axis represent light and dark periods during the day. So you can see that LHY gene here has maximum expression at the dawn or right before the dawn, while Gigantea or GI gene has maximum expression later in the day, later in the afternoon, some 8 hours after the dawn. FT protein that we have talked about is in Arabidopsis part of the output of the circadian clock, meaning that its expression is influenced by oscillations in the expression of central oscillator in Arabidopsis. You can see here that it has diurnal expression in Arabidopsis, and that on short days its expression is baseline and low, and on long days it has a maximum expression at 16 hours after the sunrise. Now, what might be the problem with our hypothesis? Every measurement so far of FT genes in olive has been conducted at 9 a.m. But since we don't know that FT expression is in olive is how it is behaving, it might be possible that if we were to take a look at the FT expression levels day to day from December to January, we might not get this picture of an increase. We might get something like this green line here. So our initial goals were to check if FT gene expression in olive has a diurnal expression. When does the maximum expression happen? And if it happens, do our previous findings that it increases from December to January still hold? So we took samples from 4 trees in natural conditions every 4 hours, starting from 9 a.m. And we took 3 leaves from 2 branches, 6 leaves in total per every sampling, from the 4th node of this year's growth, like in these pictures here. So here are the answers to some of our questions. On the horizontal axis are marked hours from the sunrise, starting from 2.5 hours after the sunrise, which is 9 a.m. actually in December here in Israel. We can see here, blue columns represent the relative expression of FT1 gene in December, and red ones are FT1 expression in January. We can see here that the expression in January is significantly higher than the expression in December. And the expression in December also has clear maximum and minimum, is also diurnal, as well as in January. We can see that the expression in December, maximum expression in December, is 4 hours later than the maximum expression in January. And maximum expression in January happens at 14.5 hours after the sunrise. Now, with FT2 results, we get slightly different picture. We see that the expression of FT2 in December is constant and low throughout the day in December, and is significantly higher in January with maximum expression in 14.5 hours after the sunrise. Now, the fact that the time of the maximum is not the same as the time when all the previous measurements so far were taken is significant, but since the expression in January is significantly higher in December, our findings that the expression rises from December to January still holds. Now, I mentioned before GI gene, and there is some evidence that at least one species, the expression of GI gene is influenced by the low temperature treatment. We can see from this paper here in 2006 that in Medicago trucantula seedlings, the expression of GI on 12 degrees treatment is more than 500-fold more expressed than on 22 or 30 degrees. Also, there is evidence in Arabidopsis that this is important because FT as GI as a part of circadian clock has the ability to influence other genes as FT. There is evidence in this paper from 2011 that in Arabidopsis, FT expression is influenced by GI. And here we are able to visualize the FT promoter by using a reporter gene that gives out the blue color. So on this picture here, we can see wild-type Arabidopsis leaf that expresses FT only in vascular bundles. But when looking at a genetically modified plant that expresses GI also in mesophyll, we can see that FT is expressed also in mesophyll. Now, the way that they think this is happening is by GI reacting with binding to the repressors of FT, and by changing their shape, it enables FT gene expression to go up. Here they provide a proof for that by showing that GI at N-terminal region is binding to all FT repressors. There are more evidence in this paper that I'm not showing that GI might actually bind to FT promoter itself directly, and therefore regulating its transcription. So after looking at our first experiments, our next question is there a chance that the expression of GI in olive could also increase as a response to cold temperature? And might that be the mechanism of flowering regulation in olive? So we looked at the results for GI expression in the same time points during the day. And we can see that the expression in December is low and constant, and in January is significantly higher than in December, and the peak of expression also appears at 14.5 hours after the dawn. We can see clear minimums and maximums, and the minimum expression is 22.5 hours after the dawn, which is just before the dawn of the next day. Now, what is interesting here is the similarity between the patterns of expression of GI and FT1 and 2 genes in olive, especially between GI and FT2. We can see that the expression in December is constant and low, and the maximums and minimums are at the same time points. Now, this goes to the favor of our hypothesis that GI might be involved in the regulation of FT expression and therefore in the regulation of flowering induction. Another gene that we looked at as a reference and did not expect to find that is influenced by the temperature, although it might be that it is the case, is LHY, also one of the genes that is part of the circadian clock. Now, these are the results from 4 trees. These are combined the results from 4 trees, and one might say that there is some pattern to it, that the expression levels in January are higher than the ones in December, and that the peak of expression in January on colder temperatures appears later than in December. But we can't draw any reliable conclusions from this data, because when we take a look at the GI expression by individual trees, we can see a recurring pattern. We can see the minimums and maximums in January and in December appearing at the same time. Now, what you can see here as maximum appearing sometimes at 18.5 hours and sometimes at 14.5 hours is probably due to the fact that the maximum actually happens sometime in between 14 and 18.5 hours. And what you can see here as maximum is actually the shoulders of the curve. But when we take a look at the GI expression by individual trees, we can't see a recurring pattern. And there might be there might be many reasons for why we can't see a recurring pattern. If you have more questions about that, I can address to it in questions section. But for now, we can't draw any reliable conclusions from the LHY expression in January and in December. What we can say, definitely, is that FT1 and FT2 expression in olive are baseline in December and do significantly increase in January. That the maximum expression happens in 14.5 hours after the sunrise, and that is the optimal time for measuring FT expression levels in olives. And that the GI expression is significantly higher in January compared to December, and it is very similar to FT expression in olive, which might suggest that the GI is required for the increase in FT during the winter. Finally, I would like to say special thanks to Professor Alon Samach and the member of his lab, Alon, Tahel, and Amnon, who was there in the beginning, to Pierce Foundation, and last but not least, to Lirit and Nurit, who have helped organize us all. We know that that's not easy. Thank you very much.
Are the practical implications guiding the motivation for this research using temperature to manipulate flowering time of flower trees? Of olive trees?
First of all, we wanted to see what is the factor that affects the flowering in olive. So when we established that it is the temperature, the regulation of temperature is not easy by itself. So it is probably more realistic to expect to find some kind of chemical that can change the effect of the temperature on flowering initiation.
There was another question there?
No, I promised them that I'm going to throw things at them if they ask, so no questions there.
That's why your rate's going down.
We saw that GI expression was high in January, I think in 14.5 hour or 18.5
Yeah,
hour.
yeah.
But we could also see the high standard error there. What was the reason maybe?
Here?
Oh, yes, yes.
Well, first of all, the difference between December and January here is significant. So although the error is high, when you do the statistics, it is significantly different from December.
Can you say additional thing about the function of Gigantea as a protein in other systems? What does it do in terms of the function?
Well,
Beside
that it binds
maybe,
to
regulating the expression.
regulating the expression of FT. They say that it might happen directly by binding to FT promoter region, or it might happen by interacting with repressors of FT.
Independent of that, yes.
Independent of that, do you know of any other function that Gigantea does?
Not.
Where is it working in the cell?
It is nuclear protein, so it is bind to the nucleus.
Do you know anything about its effect on the protein degradation or anything?
No, sorry, not at this.
Any more questions? Okay. So our next speaker is Dorcas, who did her project with the yard here.
My name is Dorcas Derito, and I'm going to present to you my work on the phenology of selected barley Heb 25 NAM families under heat stress and the genetic variation underlying, and under the supervision of one and only Dr. Yal Friedman. And my scholarship was sponsored by the International School of Agricultural Sciences. So now I want to introduce to you about this plant, barley. Barley is one of the most important cereal crop in the world. It is number 4 and used for human consumption and as an animal feed. Now, as we all are aware of the climate change that is happening, the trend is to the increase of temperatures, meaning that the current crop is being challenged by the heat stress, which is a serious abiotic stress that is challenging production of various crops, and barley is not left out. So in the domesticated gene pool of the barley, this abiotic stress, we will only be able to overcome it if we can introduce genes from the wild, because the Hodium vulgare, which is the domesticated barley, is able to cross with the wild barley, which is the spontaneum. And by doing that, the only way to do that is to create a population where we can now mine and be able to link and associate the genetic variation to our quantitative traits that we are interested in. And one of the ways is by use of a nested association mapping, simply NAM population, which is a multi-parent population. And you can use that to now get the required, our candidate genes to improve our crop. So I will describe here the population from which we selected our lines to work on. So the population we are working on is known as Hale exotic barley. It was developed in Germany by a scientist called Klaus Pilen, and it's a cross between a German spring barley called Barki and 25 other wild donor accessions, mainly from the Fertile Crescent and Tibet. So what they did is they did an initial cross between the spontaneum, the wild barley accessions, and the elite cultivar. And then in the F1, after the F1, they back-crossed to the elite parent. And then after the back-crossing, they did 3 selfings to get a total of 1,420 lines, then divided them into 25 families. So then after that, because I said we want to link this population to the genetic variation that we have, so we need a marker system, and that involves genotyping. So what they did is they genotyped the parents and these 1,420 lines, and they used the Illumina iSelect 9K chip, and out of the SNPs that they got, the 7,864, only 5,398 were informative. So basically this is what we used for our mapping. So then we come to how did we select the lines to use for our project. In an initial sort of some accidental exploration, because it was an off-season propagation under heat stress, they discovered a heterozygous Gigantea locus that was giving higher grain number, and also an experiment under drought done by a previous student. He discovered a locus known as Hodium spontaneae dry 2.2, and this locus was giving a conditional effect on grain yield and the grain number. So these our 5 families that we selected, they were either segregating for the GI locus, for the flowering genes, that is VRN1 and VRN3, and non-shattering. So now we set our goals, and our main goal was to identify favorable QTLs that could help us improve on barley heat stress tolerance. Also to build heterozygous markers for heat stress condition loci, and to determine the yield of different genotypes under optimal and heat stress. So how did we go about it? Our experiment we conducted in Arava between the period of December last year and June this year. We had, I said before, 5 families and 224 lines. Then it was a net house experiment, 8 plants per plot, replicated 3 times. So now when we come to our treatments for the heat stress, we covered the net house with a polythene bag to maintain the temperatures high, and left the other one uncovered. And by that, we were able to maintain relatively high temperatures, around 20 for the heat stress and around 15 for the no stress. So then we went back to the field to record on the traits that we wanted to study. And one of them was the number of tillers, then of course number of spikes and spike weight and grain weight. So as I said before, our lines, part of them were segregating for the flowering genes. So we wanted to follow the flowering dates, if they could be changing with the heat stress, and also we wanted to look at the plant height. But that was impossible with time, because as you look at this here, during the young stages of the plant, you could differentiate between the plants. But with time, when we went back to the field, it was almost impossible. I just don't have the picture, like the advanced picture I would have shown you, but it was impossible to tell the difference between one plot to the other. So we did not collect data on flowering dates and plant height. So then, since in my introduction I told you we discovered that there is a heterozygous advantage in the GI. Now we wanted, as part of this project, we wanted to select markers that could help us to do a backcross for the large proportion of our population. So what we did, we went back to our markers system, the markers structure file that we had. And out of 5398 SNPs, we selected markers that could represent our lines in their wild genotype. So then that gave us 7 representative SNPs to represent 205 lines. Then we designed markers for that and ran a PCR against with the 2 parents, that is the wild and one elite, and also we did an F1 in the lab and also ran a PCR for that. So how do we know now? How do we know which markers give us polymorphism? Because that is what we are interested in. So after that, we did a post-PCR analysis known as HRM, high resolution melting. That is basically tracking DNA melting in real time. And these are the results. Out of the 7 markers, 3 were polymorphic. And to mean polymorphic is to see that we could differentiate between the F1, the Barkey, which is the elite parent, and the wild parent. And to your y-axis, this is the fluorescence, how the fluorescence changes as the DNA melts, and this is the melting temperatures. So these 3 polymorphic markers were able to represent 81 headlines. Then what do you expect when the markers are not polymorphic? These are the results. So out of the 7 markers, 4 were non-polymorphic. These ones, we cannot use them for any selection. And if you want, I can show you the rest of the melting curves, but I didn't think it was important because this represents almost the whole picture. So then how did the phenotypic, like our main phenotype, change under heat stress and no stress? So we found out, as expected, the number of tillers were decreasing under heat stress significantly. So, and this could be tied to the fact that under heat stress, photosynthesis is compromised. And so we are seeing it as a reduction in production. So then our goal was to discover QTLs that could help us improve our elite cultivar. So for this, we did a genome-wide association study on tassel with a maximum of probability of 0.05 and 1,000 permutation to reduce the errors. And what you see here are Manhattan plots from the tassel software, associating our SNPs. These points you see here are the SNPs, and this is the physical position in the chromosomes. Barley has 7 chromosomes, and to your y-axis, this is the probability of associating the SNPs to our trait. So under the control, we could find several significant SNPs, and okay, we said these are our QTLs, because we said anything above 3.8 p-value is significant. We can term it as a QTL. So now we wanted to see, okay, we have the QTLs. How are they interacting with the environment? And again, apart from being, how is the genotype at each QTL interacting with the environment? So for that, we did a mixed model and developed reaction norms for our QTLs. And what we find out is that, okay, these QTLs you see here, these two are the ones that gave us a significant Q/A interaction in the control. And what is evident here is that the effect is reducing in heat stress. So now what happens in the heat stress itself? And we got very interesting results, because one of the QTLs that gave us a significant Q/E was also giving us an advantage G/E in the heterozygous SNP loci, I mean. So, and that is good to say that this is an interesting loci that can be used to improve our elite cultivar, because first of all, the G/E interaction was significant, and we had a large number of lines in that locus. So then, under heat stress, we wanted to see how, if we can tell from the beginning if our early phenotype can project what we will get in the final phenotype, like in the yield. So here what we did is we correlated the number of tillers per plant against spike number per plant, and also number of tillers per plant with the spike width. And what we found is there's no correlation. So this could be explained in the fact that under heat stress, probably some of the tillers did not undergo successful pollination and grain filling, and that's why we are getting a disconnect in the correlations. So then, since this was a stress experiment, we wanted to do an overhaul of our stresses that we had done for these particular lines. So previously, there was a drought experiment done in our lab, and we wanted to correlate to see how our current heat-stressed lines are performing against the previously well-watered, and also to see if we can get lines that are performing well under heat stress and also in the drought stress. So here what we said is anything, okay, these are the ratios. As you can see, these are the ratios, and to the y-axis is the number of lines that are represented in each ratio here. So we said any line above 1.5 to the right is a good line according to us for the stress index. So, and out of these lines to the right, 1.5 to the right, we found these 3 lines that were really performing very well in both dry and heat stress. So then we went back to the circadian clock. Initially, I talked about the gigantea. When the gigantea was discovered, they just thought maybe it was just a gene. But then when they went back to the clock, they saw that it was related to the clock. So now for this trait, we wanted to see if our lines are correlated to the clock and how it is changing under stress. So what you see here are 15 genotypes that we studied under heat stress and under normal temperatures. And then the correlation between 2 parameters of the clock, that is the amplitude and relative amplitude error. So even though we did not find any correlation, because we had like only 3 replicates, we think this is an interesting trend. Like you can see that the number of tillers are increasing in heat stress as the same as the amplitude is increasing. So these are interesting trends, and if we can increase maybe the number of replications, we will be able to get a significant, or maybe understand more the clock. So then, in conclusion, chromosome 1, 2, and 5 we found that they are the most interesting in influencing the QTLs that are influencing our tillering in barley. So then, in the next course of experiment, we want to add more traits, because tillering alone, we cannot rely on it, because most of the time it's characterized by low heritability. Then we can also say that the circadian clock parameters, the amplitude and the RAE, seem to be influencing productivity under heat stress. So then in conclusion, the Hebner population is a highly varied population with genes that could be useful for barley improvement. And with that, these are my references. I want to acknowledge my mentor, Eyal Friedman, Lian Meshuk, Hale Bisharat, and Roy Sieberman. They helped me come up with the results and, help me in everything that I did in the lab. I'm forever grateful. I want to acknowledge the Hebrew University and more to the International School of Agricultural Sciences. And with that, thank you very much.
How did you measure the changes in amplitude of the clock?
That one we didn't do in the lab. We had somebody do it for us in Jerusalem. But in the next phase of what we are doing, we have a machine now. We will be able to do that.
So do you know how they do it there? Or?
No.
Any more questions?
So do you have any idea about candidate genes in those regions that you identified?
I tried to compare my work with the previous work done, and I could not find any related gene. But although I could try and maybe blast it and find out what these genes could be doing, but unfortunately I haven't done that. I will do it.
You mentioned that in the beginning that the GI, when it's heterozygous, is giving
Yeah.
improving yield.
Yes.
Correct. So,
Yeah.
what hypothesis would there be that the gene, when it's heterozygous, is giving the highest yield? What would be the other examples, and how could you explain this?
I don't know, but I would explain it in terms of heterosis. Maybe the wild that is coming in is giving an effect to and giving a boost to the elite allele. So that's how I could just explain it. I don't know.
Any other questions? Thank you.
Good afternoon. My name is Nathan Jan. I'm presenting on the topic biological roles of ABA receptors, especially the activation of ABA signal by specific receptor in plant tissues. My supervisor is Dr. Asaf Moskuna, and I'm sponsored by PS Foundation. The main challenge of climate change to agriculture now is many events of extreme temperatures. And this situation with increasing in temperature and limiting water, there's a drastic effect on crop production. But how do plants respond to this stress? It's by the production of ABA. And when plants do not produce ABA, the result is wilting, as seen in the mutant. So let me take you through the ABA pathway, the proposed pathway. So when there's no ABA, the receptors are inactive, thereby releasing the phosphatase. The phosphatase bound to the SNACs by inhibiting it, and then we don't see any phosphorylation of downstream target proteins. But when there is ABA, the receptor is active, inhibiting the phosphatase, releasing the SNACs, and we see ABA response either by activation of ion channels in stomach culture or transcription factors in gene expression. In our biopsies, there are 14 different ABA receptors. And some of these receptors are redundant. The redundancy means when there's a loss of gene, we don't see any visible phenotype. So how do we activate a receptor? In the normal condition where there's ABA, the receptor is active, which inhibits the phosphatase. So there was a receptor which was engineered in the ABA binding site, and they found mutations, and these mutations conferred on the receptor the conformation where it mimics the ligand occupancy by stabilizing the interaction as if there was ABA. So with this mutation, the receptor and the phosphatase act as if there is ABA. So in other words, they increase the basal activity, which is constitutively always active. So when there's a constitutively active receptor, the pathway changes. But without or with ABA, the receptor bounds and inhibits the phosphatase by releasing the SNACs, and we see the ABA response. That's with the constitutive receptor. So in this research, our receptor of interest was PIL4. So I compared PIL4 to another receptor, PIL2, for us to see the distribution and the intensity of the expression. So in PIL4, we realized the distribution is much higher in the leaves, and in PIL2 is in the roots. And with this, the intensity is much higher in the PIL4 than in PIL2. The overall meaning is that different receptors have different gene expression pattern. So our hypothesis was, we hypothesized that individual receptors regulate different biological functions. And our main was to understand the biological roles of ABA receptors, specifically to identify in what plant tissues ABA signaling is activated by the PIL4 receptor. So to explain the ABA signal reporter line, when the snacks are released in presence of ABA, we realize ABA activates many genes. And out of this gene, one of them is the MAPKK promoter, which we fuse it with gas as a reporter line. So how did we answer our question? We had plants with a native promoter, which is driving our constitutive active receptor, PIL4, on the background of no ABA, because we don't want the endogenous receptors of ABA to be activated. And we crossed it with another plant, which also on the same background, but with the MAPKK18 promoter driving the gas. And we crossed these two plants. And our progeny, we believe to have both cascades from these two plants on the same background of ABA, no ABA. So when we got our F1 seeds, we grow them on MS in a controlled chamber for 10 days. And after the 10 days, we placed them on media, on MS media for the treatment, and some was also on for other stress treatment. And later we did the gas staining to see the expression of the gas gene. But we incubated it overnight. So this was our results. So for our control, that's the ABA deficient plant driving the MAPKK, with the MAPKK promoter driving the gas. We observe, because of the minus ABA, there's no ABA, which renders the receptor inactive, and there's no gas expression. So we decide to put the same plant of the same background under a little bit stress. So we place them on 0.4 sorbitol for osmotic stress, because it's believed stress activates. But we realized still the receptor was inactive, thereby there was no gas signal. So we did a positive control by exogenously applying 10 micromolar ABA to our same plant of the same background, which is of no ABA. And we realized there was a gas signal. So we compared our receptor of interest, which is the PIL4, to our control. And our control, as shown earlier, there was no signal. But our receptor, which is our F1 generation, which had both cascades, we realized by the addition of the PIL4, the constitutive receptor, the gas MAPKK promoter was activated, and we saw the gas signaling. So we decided to look on tissue specificity. where in the plant the signaling is activated. So with our control, in the seedling, we look into the seedling and in the stomata. In the seedling, we had no activation in our control. And our positive control, we observed with 10 micromolar ABA for 1 hour, there was an expression in the roots and in the vasculature. So we said, and the same thing happened on the stomata. But our F1 generation, which had both caskets, we saw the expression of the signal in the roots and in the vasculature. And also, we saw it strongly in the stomata. So summary, we know ABA activates many genes. But with this research, we realized our PEL4, our quantitative receptor we used in this case, was able to activate our gene of interest, which was the MAPKK18. But in future prospects, there's a need for us to understand if a receptor activates a subset, and in this case, all these blue lines you see here represent different genes because ABA activates many genes. So in future, there's the need for us to understand if a receptor activates a subset of the genes or all the genes. Thank you. So we also have to cross, possibly to do phenotyping in future and see how the degree of phenotypic change among these different receptors. And if possible, we can cross different receptors and see how we fared. I would like to thank Dr. Asaf Moskuna, who has been of great help, and thanks for his patience. And my senior members of my lab, Oded, Rita, Gil, Weisglass, Shoshani, and Zimran, and Rita, and the International School, and Pierce Foundation for helping me with all the financial obligations, and especially Nurit and Moshe for helping me on that fateful day when I was stuck in Turkey. Thank you.
I want to ask, you check the plants in the age of 10 days?
Yes.
Can you deliberate, or maybe, from the literature, if you go further, if the plant develop in later stages, wouldn't you find similar response in terms of whether does you have the expression and
Yes.
physiological parts?
Yes, this was on the other side of the research. We tried to take the seedlings beyond the 10 days and to see the expression in flowers. And there was one which was not recorded here, but we saw the signal in the flowers. So when we take it up, as the plant grows, definitely we may see the signal, the activation.
You indicated stomata in your slide. Have you seen anything in the stomata?
In the stomata, we realized that it was in the, you could see, when you look closely, you could see in the gas cells of the stomata, that was where the activation was.
Can you explain to us again what explains the difference between this and this?
Okay, so the ABA, the difference is the distribution and the intensity of the expression. ABA activates many genes, and in this case, we only used PEL4. So we realized you could see even in the roots, the intensity of the activation by the ABA is very much higher than the PEL4. But in the stomata, we realized with the PEL4, the expression was much higher when you look closely to the stomata of the ABA. It's like as in the gas cells.
expression in other tissues here that's not here is what?
Come again?
Here we see expression in other tissues, right, that we don't see here. And the reason is, what is causing this other?
Because we know AB activates many other genes. But this,
When you say many other genes, are you talking about receptors or many other genes? I'm not sure.
receptors. It activates many receptors.
Okay.
So in this case, with this particular receptor, which is PIL4, now there's the need for us to see if this is the only receptor which can activate this particular promoter. So we have to understand, we have to expand the project and see on other levels. So for now, we cannot say this is the only one or not.
Okay. Is there any other way of showing that it's not the only one that's activating this?
Maybe we try.
The KKK.
So maybe we try a different receptor and a different promoter.
If you take a plant that has a mutation in this receptor, is there a plant like this?
None, I know.
Okay, thank you, Jason.
Thank you.
Now we get to hear Kunta, and who did his research with OE. Please, Kunta.
Okay, my name is Srinivas. From the past 10 years, I was working under Dr. Roy Ben David in the Aero Volcani Center on the project Phenotypic and Genotypic Characterization of Israeli wheat land races. Coming to just a small background about the wheat. Wheat, it belongs to the genera Triticum aestivum, the bread wheat, which the production, the consumption, the overall production consumption is something like 95%, the bread wheat. And coming to the other is Triticum durum, the durum wheat, which correspond to 5%. When we look into the Mediterranean basis, the most dominating one is the durum wheat, and it is not only just Mediterranean basin. When we see on the overall picture on the global level, it's most widely taken cereal crop. And coming to its domestication and process, when we look into the there are during the domestic process in the wheat, there are two bottlenecks. One during the early domestication, where some of the variations was were lost, and the other in the during the development of modern varieties. So here we can see most of the variations were lost, but and the loss in these variations is termed as domestication syndrome. And here in the modern varieties, they were focused on maximizing the yield potential. By maximizing the yield potential, we lost most of the variations. So we got maximum yield potential, but with the current climate change and all, we could not cope up with the the different stresses like abiotic, biotic stress. So there is a need to reintroduce the lost variations into these cultivars so that they will boost, they will maintain the same yield with coping up with the current changing conditions. So when we speak about variations, we need to look for the material. So when we look for the source for the variations, it is the one is wild species and the other is land races. So our focus is on land races. So whatever may be, and when I spoke about land races, I took land races. We choose land races because it has high locally adaptability as well as variability among themselves. So this makes us a source for variation. So when we speak about this material for variation, we need to go back to the site of origin because it is a source where you can find huge variation. So wheat was originated in the Fertile Crescent. So you can see the So you can see the Fertile Crescent here, and even Israel was also part of the Fertile Crescent. So we can also see some of the land races even in the Israel. So if you see into wheat scenario in Israel, Like once before the beginning of the last century, the dominating one was the durum wheat with slow, there was with the Zionist movement and all, there was slow replacement of the durum wheat to bread wheat, like introductions. Then with the subsequent green revolution, most of the durum wheats were replaced by bread wheat. So we hardly see here and there like durum wheat. So, but we feel like, okay, the local durum wheat here, what we were speaking here, we consider to be the land races because they were before the green revolution. So it is almost lost now. So, but we can get this material in the source for this lost or forgotten material is a gene bank. So when we speak about gene banks, for example, there is a Vavilov gene bank in Russia. This is a big gene bank. It has a huge collection of different crop plants, and it also has a collection of wheat which was collected by Vavilov. He collected wheat land races across the globe and even in the part of Israel. So, and it was collected in the beginning of the last century, and there was a continuous propagation of this material for different purposes, and they were preserved in the gene banks. So during this process, different gene banks follow different process, and during this course, most of the heterogeneity of the material was lost. Coming to the Israel gene bank also holds a collection of Israeli wheat land races, which was collected by a guy called Matatia. Maybe some of the Israelis might be knowing about him. 2 months back there was an article about him. He collected wheat, he believed to be, there are some traditional growing durum wheat, so he collected them in the locally, like traditionally growing agriculture fields of local Bedouin and Arab fields, and he just submitted to the Israeli gene bank. Okay, this was done after the 10 years after introduction of green revolution cultivars. So this was much late after the green revolution. So, and coming to this material, this is like, it was collected somewhere in 1980s, and it was untouched for something like 96, and there was a group, Beharav et al. From the Volcany, they took the material and they just propagated for one cycle, and they just took plant height and days to heading, and they were working for the rust evaluation, and they did not characterize the material whether it is clearly authentic land race or like it has variation. So they just evaluated for rust. So much work has not been done on this material. So we thought of doing some characterization and telling like whether it is really authentic and show some variability. So with this hypothesis, we thought, okay, so with all this we made a hypothesis that okay, it was collected late after the green revolution, but we still believe because it was collected from the local traditionally growing farmer fields, so we believe that the material to be authentic, and it has high variability when compared to the other collections like Vavilov, because Vavilov collection also was collected in Israel and it holds the material from Israel. So we thought of comparing with that material and with the Israeli cultivars. So coming to the overall goal of my project is to characterize them phenotypically and genotypically, and my sub goals are like, this is a material from the gene bank. First thing is like propagation and to check whether the material is authentic, and the second one is to characterize it phenotypically and genotypically. Coming to the materials, I took 3 materials. One is the Israeli cultivars, and the second one is Matatia collection, and the third one is Vavilov collection. In all the 3, there are 2 types of wheats like Triticum durum and Triticum aestivum. The proportion is different, but it includes both the wheats. The experiment was performed in a net house in the ARO Volcany Center, and it was it was designed in a 0.5 meter. You can see here. This is a 0.5 meter block with 3 rows. It was an unreplicated experiment, and apart from the annual rainfall, irrigation and fertigation were provided based on the plant developmental stage, and we had huge weeding problem because they were closely associated. So we were doing manual weeding on that. So coming to my characterization, like phenotypic characterization, I divided them into two, like vegetative and reproductive. When it comes to reproductive, it includes third leaf width, leaf cover, chlorophyll content, canopy temperature, plant height and biomass. Coming to the reproductive, they include flag leaf area, days to heading, spike length, number of spikelets, 1000 kernel weight and grain yield. Okay. Coming to the genotypic characterization, it was done based on the LGSP CASP SNP genotyping assay. Once we got the data from the genotyping, we just analyzed the data using GeneAlex software. Coming to the statistical analysis, the statistical analysis was performed during the GEM software, and all the data was analyzed on a descriptive statistics mode to calculate the variance and variable distribution. Okay, coming to the results section, this was done. This picture shows variability among the line in a collection within itself, showing different. You can see the height variation, like there are small, there is medium, tall, and you can see some. If you see closely, there is some variation even the spike. So this shows that there is a high heterogeneity among a line. So if we see other trait like spike composition, you can see high heterogeneity in the spike composition just within a line. And this is before the harvest, and this is after harvest. So based on these two traits like plant height and spike composition, I just tried to make like to see how heterogeneous the two collections are, like one which is collected much before the green revolution and one which is collected after the green revolution. So when we look into this picture, it shows how heterogeneous the Mathatia is and how homogeneous is the Vavilo is. I don't say that Vavilo is not like landrace, but it became homogeneous. It became homogeneous in the continuous process of propagation. So, but this material is still fresh and it has high heterogeneity. So, this we can use. Sorry. So, this tells us the material is worth for studying, because there is more like more to be excavated from this material. Oh my God. Okay, and I just tried to see the distribution of the different traits which I was mentioned earlier. And on a general picture, I have seen high variable distribution with all the traits. But when I look into some traits which I found to be interesting, like canopy temperature, if you see here, the highlighted ones are the cultivars. So if you see here, there are like in between like 17 to 21, but there are even lines from the collection which are falling behind these cultivars, which are to be like controls. So this is somewhat interest us, because we expect the plant to be maintain lower canopy temperature. So this we found to be somewhat interesting. And coming to the other trait, like if we see here the plant height, the modern cultivars are like semi-dwarf, and there are even some dwarf. So we expect that our land races to be tall. So if we see this distribution, it is clearly understood that the material is not from the modern breeding programs, it is something different. So if we look into another trait, the grain yield, we can also see the similar fashion, because if you take the cultivars were bred for the grain maximum yield potential. So we expect them to be on the higher side. So it is obvious they are on higher side, but the land races are lagging behind in the grain yield. So this picture gave us a clue that okay, this material is something different from the cultivars. So after that, we tried to see on a general picture how much like the Mathatia collection, Vavilo collection and cultivars are different from each other based on the phenotypic traits. This was done based on the coefficient of variance, because all the traits are in a different measurable unit. So we took the coefficient of measurement as a new read in building this radar chart. So if we see the overall picture, the orange one is the Matatia collection, the blue one is a cultivar, and the grey one is the Vavilo. If you see here, the Vavilo is somewhat widespread, Matatia is somewhat widespread than the other two like Vavilo and cultivar. So this gives somewhat a picture that Matatia collection has somewhat high variability and it is somewhat different from the other two. But when we look into Vavilo and cultivar, they are somewhat closely related, but there is some difference among themselves. But if we look into some trait like canopy temperature, like Vavilo has more variability than the cultivar and the Matatia collection. But in general picture, if you look into this, it is understood that the Matatia collection has high variability among the two materials. So after that, we tried to see the correlation between the different vegetative and reproductive traits among the collection and the cultivars. So it gave us different significant results. Like some are more significant, some less, and some are insignificant. It's in general picture. But if we look into some traits, some combinations, if we correlate with canopy temperature with the grain yield, it is negatively correlated and highly correlated, and this also it also correlates with the information in the literature. Coming to the other, like canopy temperature and biomass, the plant, it is also like negatively correlated and highly correlated. So these two are like it's like what we expected like from the literature, and the other is like flag leaf area and the grain yield. So it is positively related and highly significant, because it is obvious that flag leaf is what the more important leaf in the like cereals, because it is a leaf which corresponds to the grain filling. So it has good correlation. So after that, we wanted to see how much variability exists among the collection and the cultivars. So this was done based on the PCA principal component analysis. The principal component analysis is it's a statistical tool which we give some input variables. Based on input variables, it develops two new components which could explain the variation among population. So you can see here, like these are the cultivars. You can see they are not widespread, they are just in a close cluster. Even this trend is also observed in the reproductive traits. They are like not widely dispersed, they are like in a close cluster. But when you see into Matatia lines from the Matatia collection, they are widespread. So this tells us that the material is highly heterogeneous and it is occupying wider space on the picture. Okay, from this picture we came to some conclusion that it has high variability on phenotypic diversity. So next we tried to see how much variability exists based on the genotypic data. So this was done based on the SNPs. So we tried to compare Israeli cultivars, Matatia collection and Vavilo collection. So based on the ploidy level, the entire collection, entire material was divided into two groups. You can see here tetraploid Triticum durum wheat, and here hexaploid bread wheat. And when we look into the cultivars, like they are clustered in a group, that's it, they are not widespread. But when we see the durum wheat from the Matatia collection, it is widespread. So this gives, even on the genotypic genetic data, the collection is highly diverse. And based on these two phenotypic and genotypic data, if we try to conclude, we made some conclusions. So first thing, as my hypothesis, the first thing is whether the material is authentic or not. To say my material is authentic, we have a result. RST allele, which is the allele which exists in cultivars, which is for the dwarfing gene. So if my material is believed to be a landrace, it should not have this RST allele. So if I see the collection lines, most of the lines are like RST2 negative. So this tells me I can conclude from this that the material is authentic because it is negative for the RST2 allele. And one more which supports my result is, like when we see into the plant height, they are all they are falling behind the cultivars. So this also gives me, this also boosts my result. Okay, these are like authentic. And coming to the other objective and hypothesis, like the variability within phenotypic and genotypic traits. So based on the phenotypic data and genotypic data, we could see high variability among the collection. And coming to this, and this is a material from the gene bank. So this was propagated after 20 years, and this material will be submitted for the gene bank for the conservation. Coming to the future, the next things we need to do is like here there is a question like how to proceed with the material. Like I showed you a picture, a pie chart showing the heterogeneity between the two collections. One was collected very long back, one was collected after the Green Revolution. So here the question we made question ourselves like how to conserve the material. So we decided to separate the material within a line, but keeping under it on the same name, so that we don't lose any heterogeneity what we observed in the Vavilov collection. So and coming to the other part, like this was a small experiment, part of the big experiment. It's called Israel-Palestine landrace project, and these lines along with the other lines, they will be propagated in the next coming season. And with some like just as an example, like canopy temperature efficacy and some others. So this material has some potential and it can be exploited in the current durum and breeding programs. Okay, coming to acknowledgement, I would like to thank International School Scholarship Fund for providing me the scholarship for my master's program, and I would like to thank PS Foundation for funding for the variance events during this master's. And this was the project, the experiment was funded by IGB, Israeli Gene Bank, and this was a part of the Israel-Palestine landrace project. And I would like to especially thank my mentor from past 10 months. He was very supportive and not just, even whenever I call, he used to reply me. It's not like Shabbat or something. So, and a special thanks to Goldberg. He is a journalist, but he was very supportive during my experiment. And I would like to thank Chandrasekhar, who was ex-PDF in my lab, and to all my lab mates and batch mates who were bearing me from past 10 months. Thank you.
Questions, please.
How many land races and cultivars did you compare in your study lineup? How many lines of the land races?
Okay, like if I take Matatia collection, there were like 66 lines, and cultivars were 5. It was short because it was like short number of control plants as a cultivars, because it was an experiment of screening. It was not, as I told, it was unreplicated, so it was very short number.
I have a question. If you want to, if your initial need was to see if this Matatia collection was a lot of genetic variability, wouldn't it be enough just to do some sequence analysis to see its variation? Why do you need to do a phenotypic variation? Can you explain to us what the
because it was also done on the sequencing, because I told even the genotyping was done based on the SNPs. So it was done, but in general, like the first thing we need to know whether, because to further go, we need to know whether they are really workable material for working. If it is not a worthable material, it's waste. So we just first, it was done like the genotyping was done, but main focus was we took was like there are many, but the RST2 gene, which is the main gene in the green revolution. So we looked into that. This gave us like picture. So maybe further screening can be done, and like most genotype, like advanced genotyping can be used and sorted out which is useful and which is.
So maybe I can ask the question differently. Let's say you have two batches, Matitya A and Matitya B.
Okay.
And in Matitya A, you see that there's a very big genetic variation when you look at SNP, and the phenotypic variation that you were testing was relatively small.
Okay.
And with the Matitya B, you see that the genetic SNP variation is not so big, but you see a big variation in phenotypes. Which would you recommend to continue?
No, because I think we need to correlate and see like how, because what sometimes, because some genetic is also phenotypically expressed, so we need to look at, because I cannot just say by maybe it's, we need to look into the picture. And
if you have to guess what's more interesting to continue working?
Maybe because the more reliable is the genetic, so we will go with the genetics.
What do you think is if you try all of these cultivars outside of the greenhouses, or maybe?
Yeah, the next what I told is the coming season they are going to do it in the field. Like there are something like 800 lines to be believed to be line races.
Sorry, did you say it was unreplicated?
Unreplicated.
Okay, so how do you like? I just want to know how you can make a conclusion from unreplicated.
Okay, because this was because the material itself maybe like maybe my mentor can tell, but I'll try to explain you. This was unreplicated because it was a big experiment, and like I just showed you, my main focus was on the Matitya collection. Apart from that, we did many screenings. So the thing is like the material is heterogeneous, so we need to take whatever it is, because it's not homogeneous. When it maybe it is homogeneous, maybe we can put replicates, but it is highly heterogeneous. So even though it was like unreplicated, there were 3 rows from which I took data randomly, because I cannot just choose by my selection like okay, these 3 are tall, something. So it was anyway, it is like I feel like okay, this is random data, because there is a heterogeneity in the population.
I would just give an addition. This was done, maybe 3 of us should have maybe expressed a bit clearer, as part of the gene bank research, and also the target was, and this was already achieved and will be completed next month, actually to submit all this material to the gene bank. This was very old material from 96 or even from the 80s, and it was not clear at all that we will even have a plot. So that was the first challenge, and they were successfully, most of them were germinated. Therefore, it was already there in the field, and this also answers our own question. If they're on the field, you might as well do the phenotyping and compare. Another issue is that you're right regarding the fact that it's unreplicated and it's a problem, but therefore, this is why I think Srinivas did not went into a very complicated ANOVA test and mean comparison. It was more descriptive, what he showed, like distribution or correlation, and also you treat, in this manner, you treat basically the collection as the 2 groups, and each sample could be actually the replicate. So if you look on the collection, the cultivar may be a small group, but then Matitya and Vavilov are a group that each one represented by, I don't know, 40, 50 lines, and for this question and the hypothesis, it's relevant.
Okay, any more questions? So thank you.
Hi everybody, so my name is Sonia Ehir Musele. My research exercise presentation was on the topic an efficient pipeline for the production of new Lysianthus variety by combining flower dipping and targeted gene knockout. This research was supervised by my wonderful mentors, Michaela and Taki. As a way of introduction, Eustoma grandifolium, popularly known as Lisianthus, is a beautiful ornamental plant from the family Gentianaceae. It is, although it is actually a new cut flower, introduced cut flower in around the 1980s, it is actually gaining ground to become one of the top 10 leading cut flowers sold in the Dutch stock market today. And its increasing popularity is basically due to the rose-like flower. Maybe that's why it's been considered as the next rose. It's a long stem, it's extended vast life. The range of Lisianthus flower color is still limited in traditional breeding, so therefore new varieties is actually being sought for. The improvement of Lisianthus now has been mainly based on traditional breeding and selection method. Genetic engineering has been, of course, we know it's a good way of improving and of improving plant characteristic. So attempts has actually been made to improve Lisianthus via transgenic approach, but the lines gotten were not, their establishment has not been very successful because of the strict regulation of transgene. So a new method, which has a new genome editing method, which has now been applied in a lot of different organisms, especially plants, the CRISPR-Cas9 system, which is capable for targeted gene modification. So we wanted to use this system to improve Lisianthus. And how does this CRISPR-Cas9 work? CRISPR-Cas9 system is composed of the single guide RNA, which is the sgRNA there, and the Cas9 complex. The Cas9 protein, they form a complex together. The sgRNA, with the help of its 20 nucleotide guide sequence, directs the Cas9 protein to the target site via complementary base pairing with the target site. So the Cas9 protein nuclease cleaves the target site 3 base pair away from the PAM sequence, and this causes a double strand break, which we see here. And this double strand break can be repaired, majorly be repaired via the intrinsic non-homologous end joining repair system of the DNA. And this repair system is error prone, so it's accompanied by mutations such as insertion, deletion, alteration of the reading frame, which can cause premature termination and eventually lead to the loss of a gene function. So we wanted to drive this CRISPR-Cas9 system into the Lisianthus genome via agrobacterium, and the common method of agrobacterium transformation in Lisianthus is via tissue culture. This method, as we all know, is very time-consuming because of the long regeneration process. And in Lisianthus, it is very complicated. It takes about 10 months to get to the rooted shoots from tissue culture, and another 10 months to get the T0 plants, and then to get the seeds. So it's really takes a long time. So we wanted to introduce a new method, which is already being utilized in other plants like Arabidopsis, the Flora Dip method. This method involves immersion of the T0 plants in fluorescence in agrobacterium suspension. Then later on, we do ant pollination and get the T1 seeds from the T0. So this method basically eliminates the regeneration process. And at the end of the day, it reduces the transformation time in Lisianthus. From there, you get, from the T0 plants after the flora dipping, it takes just about 3 months to get your T1 seeds. So the main aim of this research was to produce Lisianthus varieties by knocking out key flora color genes in the Lisianthus anthocyanate pathway by combining CRISPR-Cas9 technology and flora dip. But basically for this research exercise, the objective was to design the sgRNA, So to design the sgRNA specific sequence for the flavin synthase and the chacon synthase gene. This gene will be targeted in it. These are the genes in the anthocyanate pathway. The second synthase is the first dedicated gene in the anthocyanate pathway. The flavonase synthase is the enzyme that codes for flavonoid, secondary metabolite. This flavonoid is like a side branch of the anthocyanate pathway, and it competes with anthocyanate for a common substrate that we see here. So reports has actually shown that if this CHS is being silenced or knocked out, we get loss of pigmentation and eventually maybe a white flower. Then this FLS, according to reports, according to the work I read on papers, the FLS mutant plant led to an increase in the anthocyanide production, and also to, at the end of the day, they actually got increased hue pigment, like red coloration. So these are the two genes that will be targeted, and then later on, the objective is also to clone the CRISPR-Cas9 construct into a plasmid vector to transform the Agrobacterium tumefaciens with the plasmid vector containing the Cas9 construct, then to transform Lisianthus via the floral dipping method. So the methodology. The plant material that was used was the Lisianthus commercial hybrid, the Excalibur Pink. This hybrid has been successfully used in Orange Chama's lab for floral dipping. It is very easy to cultivate, and it has a very strong stem, and also medium-sized doubled flower. So we also went further to design the sgRNA. Most of my methodology will be incorporated in the result and discussion. So I just wanted to do it like that. Then we went further to amplify and sequence the target knockout site of the FLS and CHS gene using gene-specific primers. Then we went further to ligate the sgRNA construct and the vector, which is the PROCS vector, for transformation. Then also, we cloned this plasmid into the E. coli bacteria. Then we purified the bacteria and transformed the plasmid into the Agrobacterium strain. Then delivered the plasmid via floral dipping method. So from the results, first of all, the FLS and the CHS gene has actually been cloned from this, and we got the cDNA sequence from this paper. So what we did, we wanted to know if we have more than one transcript of these genes in the Lisianthus genome. So we blasted the cDNA sequence against the Lisianthus ESCS database in NCBI webpage, and from our results, we found out that there's only one expressed transcript of FLS in Lisianthus, but we saw 2 ascension numbers of CHS. But after we aligned the sequence of both ascension number, we found that they were both alike. So maybe we have just one functional CHS gene in Lisianthus. Then we went further to further evaluate the efficacy of this CRISPR-Cas9 system in Lisianthus. We went further to design the sgRNA construct. So before in order for us to design this, the 20 nucleotide, that's the guide RNA sequence, we made considerations. First of all, the DNA sequence should be very gene specific, so that in order to avoid off targets other off target, like alteration of other genes in the Lisianthus. Then we also went further. Presence of the protospacer adjacent motif PAM sequence of 3 nucleotides in the target genome site. This PAM sequence is actually for target recognition by the Cas9. The Cas9 will not be able to cite or the Cas9 will not be able to cite the target gene or the target sequence if the PAM sequence is not present, and they will not be able to cleave that target site if the PAM sequence is not present. So the PAM sequence is very important. Then also the first nucleotide of the 20 nucleotide sequence that was designed should have a G. This is very important for the sgRNA transcription via the RNA polymerase theory. Then also we made sure that the sequence that was designed had a high GC content. This was done. This is because of course GC pairing has a more stronger hydrogen bonds, and that will help stabilize the duplex that is formed between this guide sequence and the target site. Then we also, the 20 nucleotide sequence also had an overlapping restriction enzyme cut site next to the PAMS sequence. This is for future genotyping of the transgenic line. Based on the loss of this restriction cut site, we'll be able to tell if the CRISPR-Cas9 activity is in where, if the transformed, the plant which has the CRISPR-Cas9 activity in there. So we got a lot of, we designed a lot of sequence, and we blasted them again against the Elysianthus ESTS database to also avoid off-target, and only the sequence that had the lowest off-target frequency from the list we got, those were the ones we picked. And for each of the gene, 2 sgRNAs, okay, these are the sequence that we got. This is for the FLX, and this is for the CHS. And you see the GC content of this was 70%, and we were able to get the one here for 45%, and also here. And the 20 nucleotide sequence also contained, this is the restriction enzyme cut site for each of these guide sequence that was designed. So these sequences were further synthesized. This was not done by us. We sent the sequence to a company, and they synthesized the sgRNA cassette of both the FLS and the CHS gene. And for each of these genes, we used 2 sgRNAs. From report also, we got that the use of multiple sgRNA can actually help to improve the mutation rate, and also for what's it called, efficient gene knockouts. So we use 2 sgRNAs, and use this composing of the U6 promoter. This U6 promoter is to efficiently to drive the sgRNA expression. Then this is the guide sequence, then the scaffold, and the polymerase 3 terminator. So this is for CHS. Differently, we targeted these genes differently. So we made the construct for this and made the construct for this also. Then furthermore, we went further to, okay, this is the result. This is what we got. We went further to amplify the target sites of the FLS and the CHS gene respectively. This was actually done in order to, and we created primers for them. This was done in order to confirm if there's the presence of intron is not disrupting the sgRNA target site. So, and from the results we got after gel electrophoresis, we found out that we have a large base pair fragment of more than 3,000. Then we further purified the bands and sent them for sequencing, and the sequence result we aligned with the cDNA sequence, and from there we found that the sgRNA site was not being disrupted by any introns. Then we went further and we ligated the sgRNA cassette into the plasmid vector containing Cas9 gene. They were both digested using the Hind restriction enzyme and the MLE1, and then this was dephosphorylated and ligated with the sgRNA cassette. So this is a schematic representation of the plasmid vector containing the kanamycin resistant gene driven by the nose promoter, the 35S promoter which drives the Cas9 expression, then the sgRNA backbone, and also the restriction enzyme, among other things. So we also wanted to confirm that this sgRNA cassette was properly and is present in the plasmid that we ligated. So we went further and we digested the plasmid using MLU and the Hind. And from our result of the gel bands, this is for the uncut, which acts as a control. And this lane 1 and lane 2 shows the presence of the sgRNA here and the plasmid, which shows that validates that the sgRNA was properly ligated to the PRCS plasmid. Furthermore, like I said, in the material method, we also transform this the agrobacterium with the plasmid, and we also validated the presence of this cassette in the agrobacterium, and using specific Cas9 primers, which, sorry, the primers were not shown here. So, and from our result shows that it was the agrobacterium transformation was successful, and then the sgRNA cassette is present in the agrobacterium. Then we went further and delivered this, the agrobacterium into the Lisianthus via fluoridipine. This picture here shows the stage of the flower that was used for transformation. Results from Orange Shaman's lab has already shown that this stage is okay and is efficient for transformation using the fluoridipine. Then we immerse the flower into the agrobacterium suspension containing acetylsyringol, sea wet, and sugar. These three components is part of the major components for the suspension, for the agrobacterial suspension to deliver to the plant. Then we covered this and to maintain humidity, and also pollinated later, like I think about 2 to 5 days later, and allowed the seed pods to grow. Then we collected the seeds. This is just a diagrammatic picture showing them, and then we germinated these seeds in a non-selection plate. This is the control, and then this is the seed from the transformed plant. And from our observation, the germination percentage were not different. Okay, so far we've been able to actually transform the plasmid into the agrobacterium. We've delivered the agrobacterium to the Lisianthus via fluoridipine. We've germinated seed in kanamycin, but we're not able to go further than this because of the time frame. So, but in future, we'll be looking into other, we'll germinate the seedlings in kanamycin, and so that then this is to show for the presence of the Cas9. Then furthermore, the resistant plants will be amplified using sgRNA specific primers, and then the restriction of the restriction analysis of the PCRO applicants in order to detect the loss of restriction enzyme sets. Then further, we will sequence these uncut or undigested fragments to detect mutations. And further, we will grow this plant, the plant to get the T2 progeny, and we will also do phenotypic characterization using HPLC. But this is what we can do, what we've done so far, and in the future we will do a lot of different work. So I want to thank the International School for their support so far, the Pierce Foundation, because they are the reason I'm here today in this place. So I want to thank my mentors also, Dr. Michal and Dr. Taki, for their supervision and their support and their parental guide. And also I want to thank Shira and Fang. They are the PhD students or postdoc students that are also helping me out with this project, and I really appreciate them for that. Thank you very much.
I want to ask some questions. I want to know actually.
Okay.
What is the product developed, difference between the product developed from genetic engineering and this CRISPR techniques? One question, because there is always debate about the GMO, and some people say that the product developed from this technique is not a GMO. One question. Another is, you said that use of multiple sgRNA is the effective tool. Is there a negative side effect or other things that if you use multiple sgRNA?
Well, okay, let me answer your first question first. The difference so far is that I forgot to mention that one of the advantages of this CRISPR-Cas9 system is that the CRISPR-edited plants, after much segregation, the plants, they don't really contain the transgene. So, but of course, the normal transgenic approach that is used, so the presence of transgene, there is the presence of the foreign DNA which is being delivered into them. So that's the one major reason about this CRISPR. But I think, of course, it's still being seen as a GMO, but I don't think it should be. Because at the end of the day, to me, it's like a normal conventional breeding, like just and kind of integration. And so at the end of the day, you get a homozygous plant that is not transgenic. I think it's okay to me. Then your second question was
multiple SCRN. Multiple SCRN. Is there a negative effect? Because you are using multiple SCRN, they may cut in multiple places. I don't know either whether they will cut or mutate or insert.
Okay.
What is the mechanism?
That was the reason I showed those considerations. Like if the considerations are properly followed, a lot of papers I've read, if the considerations are followed, that the PAM sequence, because these are the major things that helps to the sgRNA to kind of for the Cas9 to cleave or to act on that specific site. But if these are not present, like for example, it's possible. I don't think it's really possible for this multiple sgRNA to cause mutation in other places, but it's possible that there will be off targets. That is not. It is not because multiple sgRNA were used. I've read the paper. I think I read a paper where it was just, I think they only used one sgRNA, and that was on arabidosis, and they also got little off target. But CRISPR-Cas9 is very accurate in most of the time, and you might not really get off target in other genes, but just on the gene that you want to knock out.
Can I ask you a question, please? You say, from your perspective, this biotechnological manipulation is like simple breeding, right? It's like conventional breeding. So, I quote you, right? So, can you explain to me, to us, please, when you get your seeds population T1, what are you doing with them then?
Oh, normally, first of all, we have to check. We check for the mutations, normal. Then later on, we self-pollinate.
You have these seeds, you have these plants.
Yeah.
And how you will continue with this?
Yeah, that's what.
Okay. Suppose you were able to introduce this genetic, not to introduce genes, to edit the genome. Okay, so how you continue? Okay,
that's what I said before. I said. Well, after these ones that I said to know, after getting after doing the resistance to kanamycin, then we'll do amplification of these resistant plants. Then remember in the sgRNA construct, this guide sequence, I showed the the restriction enzymes. This would be used in order to check for the presence or the activity of the Cas9. Then we will grow this plant, then self-pollinate it. We'll self-pollinate the plant and then get the T2 progeny also. I think from there, it segregates. We get different, because you don't get, you get different mutations, and you get, from some paper I read you get different heterozygotes and homozygotes, and later on with subsequent generation you finally be able to get an homozygous plant, which is non-transgenic.
So actually, what is your final aim?
For this project or for this research?
For this research.
Oh.
No, I didn't ask this question. Sorry.
Okay, for this research, what it was just like a proof of concept. We wanted to just be able to develop and to develop an efficient CRISPR-Cas9 system that can work for Lysianthus. And if it works, it will go a long way for us to be able to improve the Lysianthus gene, other genes. So I think that's just the major aim of this project, basically. And though we will get phenotypes that are of different colors, which is good, of course, but we also want to go further, because we will go further and see if we can also do other things.
You were showing that you were already growing these seedlings.
Yes.
From past experience with Rhizianthus and flower dipping, how many seedlings do you have to screen in order to find a kanamycin-resistant plant? What is your expectation?
Okay, it's a lot. Like I would say, from the agrobacterium method, it gives, I can't remember, my mentor said it one time, it gives about 1%, but with the floral dipping, because floral dipping is actually trying to target, with the floral dipping we get about 5%, about 5% germination. So, but each...
Germination or kanamycin resistance?
Okay, but sorry, about 5% kanamycin resistance, sorry, not the germination. That's, yeah.
Okay, any more questions? Thank you so much.
Good evening, everybody. My name is Armando Garcia from Mexico, and I did my research at Professor Rina's Kameneskrit lab. And my project was the development of a protocol for rapid garlic propagation via root tissue culture. Okay, let's start with the introduction. The garlic is a monoticotyledonous plant that belongs to the family Amaridaceae and is considered as a Allium sabitorum. This plant for ancient time has been used for its culinary and medicinal properties. Also, traditionally and commercially, this plant is propagated vegetatively by sexual steroids, mainly by the use of cloves. However, there are some wild types that they have the capacity to produce flowers and seeds. And also, the fertility of this plant recently has been restored. The problem with the vegetative production is that it's very slow, and we can transmit from one generation to another diseases. Here we have the comparison between a normal garlic and a virus-free garlic, and we can see that the difference between them is enormous. Today we have some biotechnical techniques that allow us to produce massive propagation and also to eliminate this kind of problems with the production of clean plant material. One of these techniques is the tissue culture. The tissue culture allows us to regenerate a complete plant from a single cell of tissue when it was grown or it was produced on the proper and sterile condition. The tissue culture, it has 4 stages. The first stage is the in vitro culture, when we generate the ex-plant and the plantlets. Then this is doing in vitro. Then we have the ex vitro plant in the hardening, when we pass from in vitro to ex vitro, and we give the proper condition. Then we have the production of the mother stock, and finally we have the production in the file. In this project, we will focus mainly during these 2 stages. Or in Volcanic, they focus on these 2 stages, but in my project, I focus mainly during the in vitro culture. Okay, there are several routes for the regeneration from a plant through tissue culture. It can be through organogenesis, or it can be through embryogenesis. Also, it can be direct and indirect through cayuse. In this project, we focus on the indirect somatic embryogenesis. For that, first we took an plant when we differentiated into callus, then we redifferentiated into indirect somatic embryo. We did the sprouting or germination of the somatic embryo, and finally we regenerate the plant. Today, several protocols have been reported for the regeneration of garlic, and they use mineral explants, leaves, shoots, roots, flowers, and top sets. It has been reported that the roots has many advantages when it's used as a plant. For example, we can harvest around 40 root tips per club, and also the root tips are potentially free of viruses. That's a big advantage. They are really easy to produce and harvest. What I mean with this, it's easy to harvest the roots than cut the stem or the stalk and isolate the meristem. And they also have a high rate of callus formation and plant regeneration. In Volcani, the mass in vitro propagation is mainly focused by the use of roots from a sprouted topset. A topset are these little aerial clumps of bulbs that are produced in the inflorescences and are intermingled with the flowers. Also, a plant has the capacity to produce around 50 to 300 topsets per plant, and they have the same genetic material as the mother. They have the ability to sprout. This is the plant that we use in Volcani, that is a topset sprouted. And they can be used as a propagation because they are a complete plant. When I arrived to Volcani, they have this in vitro protocol that consists in the following. First, they took the explant that it consists in make several sections in the root elongation with do any differentiation between root tips and root section. After that, the root sections and the rootings were put and grown during the callus induction media and callus maintaining media with the purpose to differentiate and initiate the callus induction. After several times, they were grown under embryo induction media and embryo developed media with the purpose to generate the embryogenesis. Then they were placed on embryo germination media and plant generation media with the purpose to generate the complete plantlet. After that, they were hardening during a chamber that we have in Volcani. And finally, they were growing, they were growing outside growing, and in this case, it was doing in a net house. Our objective, in few words, our objective is to develop the higher number of plants in the shortest time. How we can do that? Well, we hypothesis that if we choose the correct explant and we put it in the same media and in the same or in the proper media order, we can do this. For this, the plant material, we use the following plant material. We use garlic clone 87 that it was a genotype developed in Professor Rina's Kamenetsky's lab by her team. And the top sets were harvested during July 16, and they were stored until 4 Celsius degree until we start this project that it was on December and January of this year. Okay, we took two different explants. The first explant, it was the root tip, that we harvest this little part of the root. Then we make other explant that we call it root section, that it was around 2 to 3 millimeters. And why did this differentiation? Well, because we believe that the root tips has a higher morphogenic potential than the root section. That means that in this part, the growth of the cells is faster than in this part. And also, because there is no connection between the root tips and the vascular basal, we can obtain potentially free virus explant. That is really, really interesting for the commercially and industrial purposes. We also have two different media order. That the first media that we call it media A, it consists in the same media order that they have at the beginning in Volcani when I arrived. And the only difference was that we put separately root tips and root sections. The second media that we call media order B, it was the same media, but the only difference it was that we eliminate the callus maintenance media, And we also put in separately the root tips and the root section. So we have two different explants and two different ordered media, giving us 4 treatments. And the first one, it was the root tips grow in media A, in the order media A, and the root tips in media B. Here, the only difference was that callus maintaining media, it was eliminated. We also did the same for root section, and the difference was the same. And we also compared between what happened when we grew it in the same media order, but we have different explants. What we want to measure here, it was the callus induction, the callus growth rate, and we wanted to evaluate the number of plantlets or plant regenerated. But we can do it that, so we do it one step before, and we measure the number of sprouting somatic embryo, and these sprouting somatic embryos in the future, they're gonna regenerate either into root or shoot, and finally into the plant. Our statistical analysis was doing by using the software JAM, and we did one-way ANOVA and two-way ANOVA to make any difference, and also a two-key stage for the statistical difference between treatments and explants. Our results. In callus maintenance media, in callus induction media, we didn't any differentiation between the medias, because callus induction media is necessary, or is the first media that we need to induce the callus. We found that there was no statistical difference between the media order, that is something that we already know, but there was a statistic difference between the explant. And we can see really clear here that in root tips, we have deformation of callus. Meanwhile, in root section, we didn't have any callus during this stage. In the embryo induction media, we found that the best treatment, it was the root section in the root tips in media A, and we found that there was no difference in the morphological stage or in the morphologic composition of the callus. All of them, in the fourth treatment, it was embryogenetic callus. However, the only difference, it was the number of callus induction in each treatment. In embryo generation media, we found that there was no statistic difference between the media order, but there was a statistical difference between the explants. And here we found something really interesting. We found that the roots that the explant that they were grown or they were taken into root section, they regenerate mainly into shoots or roots. And on the other hand, or opposite to this, we found that the root section, they sprouting and they regenerate mainly into roots. We can see here the difference. This is a shoot, and here we only have roots without shoots. That was really, really interesting. Okay, these charts represent the whole stages during this project, and I want to make focuses only in this. And we can see from callus induction media until callus maintain embryo development, we have a constant increase in the callus grow. The callus always was growing. However, when we put it, into embryo generation media and plant generation media, we can see a decrease in this rate. We did around 300 subculture, and in this we found that we have 14% of contamination. And the interesting here is that our contamination, or the highest contamination that we have it was during the plant generation media. Okay, summarizing data, we found that with root tips, we can harvest around 2.5 plants per top set. And with root section, we can have around or more than 8 explants per top set. However, we found that even though that the root tips are produced in less quantities, they have a higher capacity to produce a higher number of sprouting somatic embryos per top set than root section. And if we want to go for in time, we found that root tips in media B, they can produce the sprouting somatic embryos in a shortest time. The difference between these two, it was that in treatment B, we eliminate the callus maintenance media, and that's for that reason we have this difference in the time. Or discussion. We found that the percent of callus induction was the same that reported by other authors previously. And we found that our percentage of callus induction in root section, it was very low compared than Sen and et al. That he did a real project really similar than us. However, the difference between Sen and our project, it was that these callus were growing for longer time, almost the double than our explants. And we found that longer periods of time, more than 5 weeks, in callus induction media and callus maintaining media, it can increase the percentage of callus induction and development. We also found that we have to be very careful when we play with times, because longer periods of times in housings, principally in 2,4-D, they can generate chromosomatic aberrations, and it's something that we don't want. We found that during embryo induction media and embryo developed media, we found something different that it was reported by Sen et al. He reported that the root section has a higher ability to produce embryosomatic embryo than us, and we found that root ticks, it has a higher ability to produce embryos or embryogenetic callus. We also found that callus maintenance media gives an advantages to the explants because they are growing for longer time during the best condition. That means hormone balances and nutrients, pH, etc. We also found that longer period of time, they can increase the embryogenetic callus, or also we call somatic embryos. And we found that we were able to have a higher formation we were able to get some somatic embryo in a shortest time that it was reported by other authors. In embryo generation media, we found that root sections are capable to produce shoots, but we found that the root tips perform better than root section in both media. We also have a lot of problems for shoot rooting when they were present in this media, and this is something really interesting because the literature says that the allelum species are species that are really easy to root, and this needs to be evaluated in the future. We found that free hormones, they can allow or they can produce roots better and faster than other media. And we found that we have a high depth of sprouting somatic embryos. We also found that we need around 4 to 5.5 months in order to generate the somatic embryo in our fastest and shortest treatment. And this is really interesting because Ferrol-Eland, in a protocol really similar than us, he reported that they needed around 18 months in order to regenerate or to produce the same somatic embryo that we regenerate. We also found that our main sources of contamination, it was due to technical difficulties. And finally, in conclusion, okay, we can conclude that an efficient protocol for in vitro micropropagation was due specifically for our genotype, that it was 87. We also found that roots from sprouted top sets are an excellent explant because they have a capacity to induce callus and they have the capacity to regenerate the plant, and also because we can have it in a higher quantities. We demonstrate that the root tips has a higher ability in induce callus than root section. And we found that the callus maintaining media is necessary in order to enhance the protocol with the main purpose of provide the correct condition for longer time. And what we need to do in the future? Well, in the future, we need to continue improving this protocol. We need to evaluate that the plants that we regenerate are true type. We also need to evaluate that our plants or the plants that we regenerate are free viruses. And we need to continue to adjust future protocols to each type of genotype or genetic line. Finally, and no less important, I want to thank to my mentor, Professor Rina Kamenetsky, to my supervisor, Herb Bernard, to the lab members, Einat, Tomer, Sahi, to Claudia Salomon or the Salomon family for believe and support young Mexicans to be here, Peer Foundation and the International School, because without them, it can be possible. Reference, and thank you. Any question?
So,
yes.
the conditions in which you got roots only.
Yes.
That was with the, I forgot, that was with the root tips?
No, with the root section.
With the root section.
Yes.
So you got roots only. What will happen to these? Did you continue to follow these plantlets and see if there was any further development?
Yes.
Do you have any ideas of what you can do maybe to get shoots from these plantlets?
Okay, that's something. We were not able to due to the lack of during the framework of this exercise, we were not able to measure during plant generation media, because in that media is when we harvest the complete plant. So we measured it one stage before, and it was the sprouting somatic, that is this one. And we found that it's very when they go to shoot, it's really easy to generate the plant. But when they go to, or when they are differentiated into roots, mainly they die. For that reason, in the graph, I showed a high increase, and then it was a decrease. That the death, it was mainly for the reason they don't have the ability to regenerate shoot. And what we can do, maybe we are in the change the media, or change the hormones, or change the condition in order to give them some advantages, yeah. Because, I mean, if you can harvest 8 root sections for 2 tubes, everybody can say, let's go for root section, we can have more explant. But in this case, we didn't find that, and that's a good question. Thank you. Yes.
How do you plan on choosing for, or like selecting for mutations, possible mutations? Do you plan like going outside the lab and doing maybe phenotyping?
Yeah, that's a good question. Well, my work is mainly to make this protocol efficiently. Of course, we can have mutation, but to be honest, I don't know how they do it. Maybe something that they can teach me the next year, and it will be really interesting. Yes. Yes, Ricardo.
Okay, of course, my question is also the same thing.
Yes.
But what I wanted to ask you is, maybe if there is only root regeneration
here
C in the D, yes. But did you check maybe the media composition? Sometimes there is a ratio of the plant growth hormones may affect which stimulates the root system.
They grow in the same condition. We think, or we believe that the difference here, it was that the root tip, it has a higher or a better morphologic, morphogenic totipotency than root section, and for that reason, we can see that difference between them. Yes. Other question?
So what do you think would be the difference between the time scale in your experiments and the ones that were published?
Okay. The difference, I think, is that what they did is they didn't do any difference between the root tips and the root section. They take it everything as a single strand that it was at the beginning. And we did the differentiation between the media because when I start, my mentor and my supervisor told me, okay, let's put it separately and let's see what happened. And we see that in the first media, we get almost 95% of callus, when in the other we have 11 and 6. And we say, okay, they have a higher totipotentiality, they have a higher ability to produce callus, and with this, maybe we can erase or we can eliminate the embryo, the callus maintaining media, because the purpose of this media is to do this. And if us is already did, we can eliminate this media. And I remember that for them, the longest period, it was during callus maintaining media, and it was almost 11 months. On 11 months against 1 month, I think that the main purpose of the main difference is the explant. And they didn't do anything. Maybe they can have...
Same plant source?
Plant source, you mean?
Same plant? Because you use a specific line here.
No. They use different. That's other reason that this protocol is just for the genotype that we have, that is 37.
About virus?
It's something that we have to evaluate in the future. And okay, root tips, it has a higher ability, or we believe that it's easier to produce free plants viruses because the vessel is not connected. And I think that other advantages that for me, I haven't worked too much with other explant, but for me it's really easy to harvest one root tip that could, how do you say, sterilize a complete plant and isolate the meristem. And the meristem is not like, it's a monocotyledonous plant, so the meristem is in the stem. It's really, really difficult to isolate the stem, and here with root tip is really, really easy. And also, for example, when you use, you can only harvest one plate, basal plate, that is the meristem, and here we focus that we can have around 2.5 per. Yes.
So I have a question. You say that in the previous paper that came out, it took 18 months?
Yeah, they have 18 months.
So don't you ask how come your supervisor gave you this project where you were supposed to finish it in? If you'll be under pressure, then everything will go faster.
And he did.
No, the idea was that let's present the result that you have, and is this in this case. I want to present maybe in the last stage that I put it just in case, the whole plantlets. I am working now in this. This is the plantlets. I am having problems with the root tips that I told you. I wish I can present this, but unfortunately, the framework of this experiment only allow us to present the sprouting. Yes.
Thank you.
Thank you.
And I want to thank you for this question. We will try mutation induction in tissue culture. Thank you.
And
I wrote it down.
Okay, we finished. I just want to say that I met you the first time by Skype in an interview, and we talked to you then, and I see you now, and I'm very, very proud. And you should be very proud of yourself for this big transformation that you went through, that each one of you presented so nicely, shows such expertise in their new field. So yeah, you should be all proud of yourselves.